All right, thank you so much for that introduction, and thank you so much to the organizers for inviting me to be a part of this amazing day. It's already been so educational. I've learned so much, and as I said to a couple of colleagues, I was grateful not to have to directly follow Dr. Burns because I really needed some time to put myself back together as a relatively new mom as well as a researcher. So I just appreciate everyone sharing their stories, and I just want to say that that's why we do what we do as researchers. So I hope that what I'm going to tell you about today will add something to that conversation. So let me see if I can figure out how this works. Yes. Whoops. So my plan for what to cover today is to sort of say something that basically everyone knows is not going to come as a big surprise to this audience, that the way people with autism perceive the world is different. And we as researchers have different tools for measuring these differences. And I'm going to try to bring home the point that the behavioral reaction that we see doesn't always necessarily tell us everything about what's happening before we get to that reaction. And we as researchers are really trying to get at sort of the full gamut of stimulus to behavioral reaction and all the processing steps that happen in between. So we'll spend some time talking about that. And then I'll tell you about some work from our lab that we've done to try to merge these concepts of perception and reaction. So I'll tell you about some work we've done on pain experience in individuals with autism, as well as a prospective study looking at the potential role of sensory processing differences as developmental building blocks for the more complex behaviors that we associate with autism. And that's kind of the second point that I really hope to drive home, is that these basic sensory processing problems, we think, have a pretty fundamental role in the more complicated behaviors that we see, and understanding those better, we hope will give us a better insight into autism as a whole. So how do we know that sensory experience is different in autism? Well, one of the ways that we know is by firsthand accounts. And so you guys are probably familiar with these two writers and public figures, both of whom experience sensory differences with autism, but describe them in really different ways. So Temple Grandin kind of has a negative spin. So she's hypersensitive. Everyday environmental noises really set her off and make her feel defensive. And you can see how feeling like that would cause one to withdraw and not want to engage in their environment. Naoki Higashida describes sort of his repetitive jumping in sensory terms that are really positive. So why do I engage in this repetitive jumping? Well, it makes me feel good, and he's able to describe what the feelings in his body that sort of make him want to engage in this repetitive behavior. So we can see how sensory differences might easily give rise to the behaviors that we see in autism. So many of you probably know this, but the DSM-5 was introduced in 2013, and it's this version of the DSM, which is sort of the Bible for psychiatrists, that first introduced sensory differences in autism as part of the diagnostic criteria. So before that, the DSM didn't really recognize sensory differences per se as being part of the diagnostic criteria. So the DSM talks about this in terms of behavioral reactivity. So there's this hyper responsiveness, very similar to what Temple Grandin talks about with the school bell being overwhelming and her wanting to disengage. There's hypo responsiveness or hypo reactivity, where a stimulus in the environment that we might expect to get someone's attention does not. And then there's this last pattern, which I'll spend probably the most time talking about today, unusual sensory interests. So these are individuals who might like to look at particular stimuli in a really repetitive way. This pattern kind of maps on to the description in The Reason I Jump of the kind of a pleasant feeling as a result of a particular sensory experience that is repeated. So as I mentioned, there's a whole process here, right? So we tend to think in terms of there was a fire truck and there was a siren, and my child covered his ears and winced and freaked out. And your brain is doing all these things in the middle there, right? So it's registering that there's a stimulus out there. It's saying, oh, there's a stimulus, it's a siren, and then as it moves into perception, it says it's loud, and it's maybe at this frequency, and it sounds like it's getting closer or further away. And then there's an evaluation stage where your brain is saying, I don't like that. I want to get away from it. I want it to stop. Or I do like it. Or any sorts of other affective or emotional evaluations, as well as cognitive action plans. So what am I going to do? Am I going to try to get away from it? What kind of a reaction am I going to have? And so this points out that we don't necessarily always know whether someone's perceptual experience can be reflected by their behavior. So these two young ladies are sisters who have a YouTube channel where they taste test different foods, and they broadcast this to kids. And you can see the sister on the left, so they're taste testing limes in this still from their, one of their videos. You can see the sister on the left, there's pretty much no question as to what her perceptual experience is, right? She's probably experiencing that as sour. She doesn't seem to like it. She doesn't seem to want to eat any more limes. The sister on the right has a much different reaction, and we don't know necessarily, does it taste just as sour to her or not? Does she find it as unpleasant or not? Her sort of other factors other than just her sensory experiences are coming into play. And so it's important for researchers to be able to make distinctions between perception and reaction. So just to kind of go over it in more detail, so reaction involves overt behavior often, avoiding a stimulus, exhibiting distress, seeking out more or exhibiting pleasure, changes in facial expression. It can also involve involuntary responses. So maybe your heart rate goes up because you're stressed by it. Maybe your brain responses are different. And so those are some of the things that we try to measure in our lab. And then to make a distinction between reaction and perception, so to go back to the lemon or lime example, here we have a behavioral reaction of facial pucker and maybe a yuck, and we kind of have an overt behavior. Researchers who focus more on perception are looking at things in a much finer grained light. So they might, instead of giving you a whole lemon, give you a drop of lemon in a glass of water and try to ask, well, did you need one drop to be able to detect that it was there, or did you need two? And if I put the one drop in a whole gallon of water, could you detect that? Can you tell the difference between 1 or 2 drops of lemon? So people who study perception are looking at things in terms of the physical properties of the stimulus itself and the way the brain is processing those properties. So going back to behavior, one of the ways that our classic sort of approach for measuring behavior is to ask parents and teachers and other caregivers about behavior. And so this is a really effective tool in a lot of ways. It gives us a great window into the everyday life of the people that we're engaging as research subjects. It samples a wide range of contexts. Some of the disadvantages are that it's hard to control. Real life is messy, and it's not always, as in the case of the two sisters, it's not always easy to tell what in the behavioral reaction we can isolate to sensory differences versus other kinds of processing differences. It also depends on recall, and we can have biases in our recall. Everybody does. And so something that is a really salient experience, like a meltdown that sort of ruined your whole day, might be easier to respond and recall and respond about than something that didn't sort of have that same emotional weight. Laboratory-based observations are kind of a solution to some of these problems. So in this kind of a situation, we'll present a toy or an activity that has sensory properties and measure reactions. So that gives us a little bit more control. It's not a daily life environment. It doesn't depend on recall, but again, it's a single context, and we're just sort of looking at the laboratory context in isolation from the rest of the person's life. But importantly, both of these require inference about another person's sensory experience based on their overt reaction. So as I sort of talked about with the lemon juice, one of the ways that we can measure perception as opposed to behavioral reaction is to have highly controlled stimuli that we manipulate. And I just put this up here to give you an idea of how people who use psychophysics to study sensory perception do what they do. So they're interested in the relationship between the physical stimulus and the perception it produces. And in this case, it's can you detect a small spot of light in a totally dark room? And what they do in this first column, they start with some level. So the numbers here represent light intensity. And they go from this intensity to 1 unit lower, 1 unit lower, 1 unit lower, and they ask the person, did you see that or not? And when the response starts to change from yes to no, they make a little mark, and then they start with a really, really low stimulus that no one can detect, and bump it up 1 bit at a time until the response changes to yes. And then they'll take an average across these trials of just the exact point where the responses start to change from no to yes. So this is very painstaking, detailed work. It has some advantages. So it gives us tons of control over the sensory stimulus and the perceptual result. And it's really easy to link to brain responses. We know a lot about how the brain processes these really basic sensory stimuli that are very uncomplicated. Got a lot of drawbacks too, though. Again, it's a single laboratory context without a lot of real-world validity. And it depends on the ability to attend for long periods of time, sit still for long periods of time, and the ability to communicate with an experimenter. So these are some of the tools in our toolkit for people who are interested in understanding sensation and perception and emotion in people with autism. So what have we learned? Well, to reiterate the 3 behavioral responses, we're going to start by talking about behavior. And the work that I'm going to tell you about was sort of gathered using parent report. So this is sort of looking at behavioral reactions. To review, the DSM defines these 3 categories of responsiveness, which I'm going to refer to as patterns. So there's hypo, and sensory interest, unusual sensory interest, repetitions, and seeking. And the group whose work I'm going to highlight in the next few slides abbreviates this as SIRS. So they kind of recognize the fact that a lot of times the sensory consequences of repetitive behavior are kind of hard to separate from each other. It's hard to separate the repetitive behavior from the sensory result of it, and so they kind of have a descriptive term. So if you hear me say SIRS, that's what I'm talking about, the sensory seeking. And then there's a 4th pattern that these researchers have looked at, and this is more in the realm of perception, although we can also get at it with parent report. We know that many people with autism have a particular strength in seeing visual details. And so in this scenario, the task is to identify the red apple. And the task gets harder when there's more distractors to choose from. It takes you a longer time if there's 20 of these different things than if there's just 3 or 4. And it gets harder when there's more different features you have to pay attention to. So in panel C, all you really have to pay attention to is shape to find your target. But in panel B, you have to pay attention to both shape and color. So here's some work kind of describing how people with autism do on a task like this. This top dotted lines represent the situation in panel C. This is single feature, so all they're sort of looking for are differences in shape or one feature. And you can see that people perform overall really well on this. On the x-axis here is the increasing set size, so more distractors as the numbers go higher. And you can see that people with typical development who have the open circles here do really well, and their performance starts to drop a little bit with increasing number of distractors. Whereas the people with autism really kind of maintain their very good performance over the set size from 5 to 9 distractors. But then the really sort of powerful thing is to see what happens when you introduce more kinds of distracting features. So if you have to look at color and shape and go from 5 distractors all the way to 13, look what happens to the typically developing group again in open circles. They go from 70 to 30% accuracy. And although the autism group does sort of drop off, not nearly as dramatically. So this is a real relative strength for many people with autism. And we can get at this with parent questionnaires with questions sort of about daily life that map onto this. So we might say, oh, you know, we've been to the optometrist and my child actually has better than 20/20 vision, or my kid is amazing at doing really complicated jigsaw puzzles. So these are the kinds of things that we can get at with a questionnaire as well. So we have these 4 patterns: enhanced perception, and then the 3 behavioral reactivity patterns that we talked about from the DSM. And here's one of the problems for us as researchers. Few people with ASD show a single pattern all of the time. Lots of people with autism have multiple patterns going on at the same time. And it's hard for us to know exactly how they map onto each other. So researchers at the University of Wisconsin are kind of working on this to understand, well, can we group people based on sort of the combination of patterns that they show? So what they've come up with are 4 subtypes. So when I say subtypes, I'm talking about groups of people now. They took about 1,000 kids with ASD and collected data at 2 points over a year. And they found these 4 patterns that we've talked about. So SIRS, hypo, hyper responsiveness, and enhanced perception is this EP here. Generally sort of tend to group together in one of 4 different subgroups. So, so individuals will fall into one of these 4 bins based on their exhibition of these patterns. So the first subgroup is about 1/3 of their sample, and they just called them mild. So they tend to score overall pretty low on all of these sensory patterns. The next they call sensitive distressed, and so this has, the first 2 are SIRS and hypo, so low on the sensory seeking and hypo responsiveness side, and higher on the hyper responsiveness and enhanced perception side. The next subgroup they identified is the attenuated preoccupied, and that's basically just the opposite of the first pattern. So they score high on hypo-responsiveness and sensory seeking and repetitive behaviors, and then lower on hyper-responsiveness and enhanced perception. And then there's a 4th category where kids just tend to score high on these sensory items across the board, and they call that extreme mixed. So when you fall into one of these patterns or one of these subgroups as a person with autism, you tend to retain that subgroup, so you tend to stay in the same subgroup over time, and they've followed them so far for a year, and they're continuing to follow this large cohort. There's also associations between these subgroups of sensory processing differences and social communication and adaptive behavior, so I'm going to talk about those next. So the 2 of the 4 subtypes, the attenuated and extreme response patterns, are associated with poor communication and socialization. So here again are the 4 different subtypes: mild, sensitive distressed, attenuated preoccupied, and extreme mixed. And you can see that attenuated preoccupied and extreme mixed have poorer performance on measures of communication and socialization. And so this was actually a little bit surprising, right? Because we have this idea that the sensitive distress subtype are the people that are disengaging from their environment because things are too overwhelming. And this suggests that may not actually be the right description. So we can also think of these subtypes in terms of severity. So the first one is mild, where most of them don't have too many sensory symptoms. The next 2, sort of, there's some symptoms, but not across the whole board. And so these next 2 are, you can consider to be kind of moderate. And then there's this extreme mixed, where they have sensory issues across the board. And if you arrange them like that and think of them that way, increasing sensory symptom severity, regardless of what kind it is, tends to be associated with higher parent stress and higher maladaptive behaviors. So to summarize this little bit of work on sensory subtyping, we have these 4 subtypes that are really sort of built mostly on behavioral reactions. They also integrate perception somewhat by sort of incorporating this enhanced perception pattern that we talked about. This is stable over time within individuals, and we've heard in previous talks about the idea of personalized medicine and precision medicine, and so some of the implications for treatment is that if we can sort of identify these stable subtypes, that we might be able to target sensory-based treatments more effectively. So now I'm going to move into some work from our lab on a very specific perceptual experience that's been mentioned a couple times already today, but we really don't know that much about in autism from an empirical perspective. So there's a lot of information from clinical case reports and parent reports that suggest that people with autism may be less sensitive to pain. So you'll hear descriptions of children who can walk barefoot through the snow, not notice when they're touching a hot stove, that kind of thing. And the DSM itself actually gives, as an example in these new coverage of sensory symptoms, an apparent indifference to pain and temperature. Well, as we know, autism is a pretty variable group of people, and so this is probably not true for everybody. But even the assumption that sort of the behavioral reaction that we're seeing, or lack thereof, necessarily reflects perceptual experience might not be right. So in individuals with intellectual disability and self-injurious behavior, we actually see exaggerated facial responses to pain that sort of are not present in individuals who don't self-injure. So that's sort of a paradoxical relationship that gives us the idea that this might be more complicated than we think. And so this is really important, and I think most of you probably know this, but I'd like to highlight kind of how we think about pain as a society, how clinicians, parents, teachers, and particularly doctors detect and interpret people's pain based on their communication ability. So, we go to the doctor and they give us a scale from 1 to 10 of how bad your pain is, and they depend on our ability to verbally communicate that, or at least communicate it in some way. Well, we know that communication, both verbal and nonverbal, is different in ASD, and so there's this possibility that a lot of the pain experienced by people with ASD might be misunderstood or go undetected, particularly by doctors or other people who don't know them very well. And so that's potentially a really important problem for us to learn more about. It could result in chronic pain going undetected and untreated. As we said before, differences in pain perception might factor into self-injurious behaviors in a way that we don't understand yet. And then some associated medical comorbidities like GI issues might also be relevant here as well. So to kind of just reiterate this, we're going from stimulus through sensation, perception, evaluation, and reaction. And those reactions can be voluntary, like a change in facial expression, or they can be involuntary, so brain responses or physiological changes in your heart rate. So when we try to measure this in the lab, what we tend to do is thermal pain, so heat or cold pain. We have a device called a thermode that heats up or cools down in a very precise way. We can set it to a target temperature, and it reaches that temperature in a specified number of seconds and holds it for a specified number of seconds. And so we can ask people to do different things. Can you push a button when you first feel the temperature change? So how good are you at just detecting a change in temperature generally? Or give us a signal when the temperature first starts to become painful. And these are the kind of studies that we're starting to do in people with autism, and leading to more questions than answers, unfortunately, at this point. But the first study on this was done by me as a postdoc, and we found evidence of actually lower pain thresholds, so more sensitivity to heat pain at 2 sites, so the forearm and the palm. This was in a small sample, though, and more recent studies that have had larger samples actually haven't replicated it. So heat pain and cold pain thresholds are here. Again, threshold is sort of just the point where it just switches over from not painful to painful. And so those thresholds don't appear to be different in this sample. They might be different, but they're not really statistically different here. So this is a large battery of tests, and the cold pain and heat pain are the ones I have circled here in red. Some of these more recent studies suggest that there's maybe some other differences, so ability to detect changes in innocuous temperature changes, so warmth or cool, or mechanical pressure. So kind of a complicated picture so far of trying to use psychophysics to understand pain processing. So one of our goals with this study was to try to look at neural responses to see if we could learn more. So in this paradigm, what we did was apply a 49 degrees Celsius temperature to the leg for a prolonged period of time, so a 21-second stimulus, which is a pretty long time to have something that hot on your skin. And most, as you'll see, sort of most people find this on a scale of 1 to 10 to be somewhere like between a 7 and an 8. And notably, our group of ASD and our group of TD individuals rated it the same on average. So just to let you know, this is a group of 15 adults with autism and no intellectual disability, matched with a group of 16 typical adult controls. So it's a fairly small sample, but we put them in the MRI scanner and looked at how their brains responded to this pain. We had an expectation of the parts of the brain that we thought would respond to this. So there's a kind of a well-established literature linking brain responses to pain in just the general population. And these are some of the regions that are involved in that. So there's this region called the dorsal anterior cingulate cortex, or dorsal ACC. There's a supplementary motor area, so these are frontal regions. The insular cortex, which is sort of here, kind of on the lateral surface of the brain, but kind of buried deep inside of it, as well as the thalamus and the cerebellum. So these are some of the regions that are part of what's known as the neural pain signature. So this is sort of a group of regions that is reliably expected to respond to pain. And pain is sort of a tricky thing to study because there's all these things mixed up in it, right? So there's the actual physical heat, what's actually producing pain. There's also a lot of meta-cognitive evaluation kinds of things going on. And so this particular set of regions has been shown to be really sort of directly affected by increases in physical heat intensity, but not as directly affected by some of the more top-down cognitive or memory processes associated with experiencing pain. So we also had an expectation for sort of how this response would look. So over this 21-second stimulus, we would expect something like this. So this is the y-axis here is percent signal change in these regions of the brain. So you can think of it as brain responsiveness. And so we kind of divided this up into an early phase, so like generally the first half of the pain stimulus, an intermediate phase, and then a late phase that incorporated the offset of the pain stimulus and a recovery period. And we expected to see this ramp up during the early phase, as followed by kind of a maintenance or maybe a little bit of a decrease. And then with the stimulus offset, there's often a little bump in responsiveness that seems to indicate some kind of processing of the absence of pain. So we'll think of these phases as early or acute pain, intermediate or sustained pain, and late or evaluation. And so what we saw, I'm going to talk about each of these phases in turn. In the early stage, we really didn't see any differences between our two groups. So the response to pain in that first 10 seconds is intact as far as we can tell in our group of adults with autism. They show robust and reliable activation in all the regions we would expect. So dorsal anterior cingulate, thalamus, insula, premotor regions. So we really didn't see any statistically significant differences between the groups here. Surprisingly, as the pain stimulus continued, we started to see really big differences. So the orange here sort of is activation, the blue represents sort of deactivation, and so you can see that there's no more orange during the intermediate stage in the ASD group. So their response to the pain is totally turned off as it goes on. And then we see the same thing in this late or evaluation stage. Again, no orange here. As expected, the TD group is starting to ramp down. Most of this is after the stimulus is ended. And so we're seeing this dampened response. This is another way to show the same data. This is a subtraction map. So 3 phases are here, early, intermediate, and late. And this kind of shows the parts of the brain where the typically developing group showed more responsiveness than the ASD group. And so you can see here, these brains are empty because the responses were pretty much equal across the two groups, whereas in the intermediate and late phase, widespread sort of increases in activation in the typical group relative to the ASD group across the pain network. And so I'd like to sort of preface this next slide with just a little caveat that it's already a pretty small sample. So the whole autism group was about 15 people. And so this is dividing that group into roughly half of individuals with autism who endorse some form of self-injurious behavior versus those who didn't. So take it with a grain of salt. It is a small sample, but the differences that we see when we split that autism group up are somewhat provocative. So the green here is the typical group, and the bars here that are kind of faint, but the 3 gray bars represent our early, intermediate, and late phase. This is in the left insula, which is a key node in the pain network we talked about. So you can see in the insula that there's a ramp up in the typical group that is sustained throughout the intermediate phase, and then this little bump that we would expect in the late evaluation phase. And then if you look at the red and the blue, this dampening of the intermediate and late response seems to be driven almost entirely by those individuals with autism who show some sort of self-injurious behavior. So that's that's something that we really want to follow up on. Because it does kind of fit with this narrative that individuals who engage in self-injurious behavior may actually have a difference in sensitivity. based on their neural responses. So to summarize this study, communication differences make understanding how pain is perceived in individuals with autism both difficult and critical. We don't know yet sort of whether on the whole there's hypo or hypersensitivity. There's probably both in different people, but we have mixed results using psychophysics thus far. We do see in this small initial study brain responses to prolonged pain kind of being blunted as the pain goes on. And so the directions that we're hoping to take this line of research is that we're going to try to link these findings in the brain back to the behavioral response, get a better understanding of some nonverbal ways that people with autism might be communicating their pain, so measuring facial expression. There's quantitative ways to do that in people with autism, and that might help us get a better link between the brain response and the behavior. A caveat here, so because of the nature of this work, we don't do this work in children, and we don't do it in people with intellectual disability. So this research is limited to very cognitively able adults. We hope that we can use that as a tool to understand the way pain is perceived in the broader population, but just the ethics of doing this in those populations is prohibitive at this point. So here's this sort of picture again, and this picture that we're trying to build of the relationship between sensory and broader autism symptoms. And so we might construct this narrative that the perception of pain, we go from the stimulus and we see that in the brains of these people with autism that there's decreased processing or decreased brain response at the levels of perception and evaluation. And so that might lead to a difference in reaction. That might lead to self-injurious behavior to try to kind of amplify signal that's not there. But there's a really important alternative explanation, right? It could be that over a lifetime of engaging in repetitive self-injurious behavior, that actually is what changes the brain responses, not the other way around. And so we have this kind of chicken and egg effect when we're looking at people who have already undergone several stages of development, as we do in adults. And so to be able to sort of tease apart what is chicken and what is egg requires a different approach. So again, the looking at this other aspect, socialization or social interaction deficits in autism, we see socialization. It involves things like responding when someone calls my name, making eye contact, using touch to express affection. And as you can see how basic differences in sensory processing in these domains might impact these behaviors and give us the sort of behavioral phenotype that we see. But again, chicken and egg, the only way for us to really establish which direction the relationship is going is through predictive relationships, not concurrent ones. And so that calls attention to the need for longitudinal prospective studies of infants. So this last part I'm going to tell you about is kind of thinking of sensory processing differences through that lens, thinking in terms of development as basic sensory processing being something that develops very early in life, systems that come online in infancy, and if something is going awry or is different, processed differently here at this very early stage, then we will sort of have an altered trajectory for the more complex behaviors that these basic sensory behaviors serve as a scaffold for. So that's the sort of premise that we're working to test in a study that we're doing with high-risk infants. So as was mentioned earlier today, infants who have an older sibling with ASD are 20% more likely to have ASD themselves. And so if we can start to understand these sensory differences before the symptoms become evident, then we can better understand this directional relationship if it exists. And part of the toolkit that we have to work on that are patterns of brain activity. So there's things that we can measure with EEG that are kind of relevant here for ASD symptoms as well as development. I'll get into those, I think, in the next slide. Yeah. So using EEG, we're able to measure lots of things, but the one that's relevant here is a pattern of resting brain activity known as frontal alpha asymmetry. So this has a particular developmental pattern, which I'll describe to you next. So it's sensitive to early brain development. It changes across infancy. And It's also been known to be associated with the degree to which someone engages or approaches their environment. And it's also been shown to relate to autism symptoms, as you might expect, given that engagement. So this is a study that sort of was done outside the context of sensory processing, but just looking at this pattern of frontal asymmetry. The low-risk control infants are here. You can see at 6, 12, and 18 months, there's this shift. Going from a pattern of rightward asymmetry, which is considered to be a more developmentally immature pattern, to 18 months, kind of showing a reversal of that to leftward asymmetry, whereas the high-risk group of infants who have an older sibling with autism show the reverse pattern. So we can already detect by pretty early in the first year of life that there's something different about the way the brain is developing as a whole there. So because of the relationship with this pattern and engagement with the environment, we've focused particularly on this pattern of behavior. So this is sensory interest, repetitions, and seeking behavior. So I'm going to try to play this video and talk over it. So you can see that this little guy is really interested in the sensory properties of this Slinky toy. And I'm only showing the short clip here. But during the course of our assessment with him, he was pretty much inseparable from this thing. He's investigating the sensory properties of it in various ways, including smelling, but a lot of this visual inspection. And so he's really, really wrapped up in this. And you heard the phone ring in the background, and we did other things during the course of the assessment to try to engage him and get his attention. And he just really wasn't interested. And it seemed like his interest in this really took precedence over anything that we were trying to do. And so the idea that we wanted to test is whether that sort of, you can imagine that over the course of development, if you're engaging with non-social stimuli in your environment in that way, that it results in decreased opportunities for social engagement, and that might sort of lead to the social differences that we see. So over the next several slides, I'm going to show you data in a group of high-risk infants, so SIBS ASD is what we call these guys, compared to low-risk infants who have an older sibling that has typical development. And it's always going to be sort of a range like this. So the black is going to be the SIBS TD control group. The high-risk group is going to be either in gray or in red. The distinction here is that the red babies are ones who went on to receive an ASD diagnosis at 3 years, whereas the gray have an older sibling with autism but did not receive a diagnosis themselves at 3 years. So you can see, not surprisingly, that sensory seeking behavior, or the SIRS pattern, is much stronger, both in frequency and intensity of sensory seeking in the SIBS ASD group, with an outcome of an autism diagnosis relative to the other two groups, but that even overall in the high-risk group, it's higher than the control group. So that's not a huge surprise. What we did see is that the SIRS pattern is associated with frontal alpha asymmetry, as we hypothesized that it might. So in the high-risk gray and red group, the higher the sensory seeking score, the more rightward asymmetry. So this is a little bit confusing the way it's oriented, but the rightward asymmetry is on this side, which is, again, the more developmentally immature pattern. So we see this association of the seeking behavior with developmental immaturity in this frontal alpha asymmetry pattern. And as seeking behavior gets less, we see the more mature pattern in the group of gray and red there. The typical group had a flatter line that didn't really reach statistical significance for a relationship there. The important thing that we found from this is that it's actually predictive. So this sensory seeking pattern in infancy actually can predict later autism symptoms. So at 18 months, if we saw higher levels of sensory seeking, we also saw higher levels of social symptoms, at 36 months when we measured diagnostic outcomes. So again, the model for how this might be happening is that attention is focused on non-social elements in the environment, which kind of just overshadows opportunity for social engagement. And then we miss opportunities for social engagement, and that leads to social deficits over time in sort of a cumulative way. So what we saw is that increased sensory seeking is actually, so we looked at opportunities for social orienting using an observational assessment where we tried to solicit opportunities for social engagement and looked at the response. And so at 18 months, we're able to measure social orienting. And we see that the higher the sensory seeking score in the high-risk groups, the higher their scores are on social orienting, meaning that they show decreased social orienting. So the relationship here is that decreased social orienting is related, as we might have expected, to increased sensory seeking behavior. And so what we really wanted to know was, what's the direction of relationship here? And so we statistically looked using a mediation model at the relationship between these three things. So 18-month-old sensory seeking behavior, there's a relationship with future social symptoms, there's also a relationship with social orienting. If our model is right, then we would be able to say that the social orienting is actually the mediator of this effect. And we were able to show that because in the mediation analysis, this effect went to 0 when we accounted for this. So what this tells us is that early sensory seeking behavior is in fact impacting future social symptoms via decreased social orienting. And that mediation was moderated by group, meaning that this whole relationship was significant for the high-risk group, but not for the low-risk group. So to summarize this part, our pattern of sensory interest seeking repetitions is more frequent and intense in high-risk siblings. And early sensory seeking behavior is associated both with abnormal EEG asymmetry and social symptoms at 3 years. SIRS appears to be impacting social symptoms by interfering with social orienting. So the compelling nature of these sensory stimuli in the environment that are non-social seem to be just getting in the way of orienting to and attending to social stimuli. But we need more comprehensive longitudinal studies to really sort of get at the specific nature of this and to understand how we might intervene on this. So how this might sort of translate into a difference in treatment approach at 18 months when we're first starting to see these differences emerge. So to summarize the talk as a whole, we as sensory researchers looking at autism are trying very hard to bear the whole process in mind. And so we use a variety of tools to do that, but we really want to be able to understand everything from the physical stimulus properties themselves to the behavioral reaction and everything that's happening in between to get a really clear picture of what's different in autism. And as we've heard several times today, there's not just one autism. There's definitely different flavors. There's different single gene models have different sensory profiles. Idiopathic autism has different profiles mixed within it. So really sort of understanding how to subtype sensory differences in autism will help us to both understand it better and to advance personalized treatment approaches. I called attention to the experience of pain, and that's a perceptual difference for people with ASD that we don't yet know a lot about, but we think that it's pretty important because communication differences may have a pretty big impact on quality of life, and so we really want to direct a lot of our attention to better understanding that as time goes on. And then finally, to get at the sort of chicken egg question of whether sensory differences are kind of, as we're thinking, building blocks that are changing the developmental trajectory of more complex behaviors, we need prospective longitudinal studies that focus on infants who are at high risk. So that is all I have. I just wanted to take a minute to acknowledge the people who worked on the data that I presented to you. So Michelle Fele is a postdoc in our lab, and she did the pain work, particularly the imaging part of the pain work. The behavioral part of it was also supported by collaborators at UNC and University of Southern California. And then the infant sibling work is a collaboration between Tiffany Wojnarowski at Vanderbilt and me, and these individuals helped us as well, and as did Wendy Stone and Cara Damiano at Duke University. So it was really a collaborative effort with wonderful colleagues, and we've had lots of great support from NIH. The Marino Autism Research funded the infant SIB study, and then both Autism Speaks and the Autism Science Foundation have funded different phases of the pain work. And of course, our biggest group to thank is the people who come into the lab and allow us to poke and prod them and ask them a million annoying questions and their families. So we're most appreciative to them. And I'm appreciative to you for your attention, and that's all I have.